right. I feel like I have a lot more power right now. All right, I apologize everyone if this runs a tidbit long. All right. In the fall of 2020, I had a very good opportunity to dissect and study a cadaver in gross anatomy. In the latter half of the semester, we did our prosections. I did mine specifically over the neck and shoulder. And what I had found was that, you know, as different as we are on the outside, we are just as different on the inside. So these differences are very fascinating, and from what I found, and they actually have real implications in surgery, PT, and other fields of medicine. So here is where I'm going to present my research and some of my discoveries, as well as the experiences of several doctors with their anatomical anomalies that they've had. So I broke this up by body system. The first was the reproductive system, the septate uterus. So this is basically, it's a congenital divided uterine cavity that only affects about 1 in 100 women, which is fairly common for abnormality. So we don't know what directly causes it, but we know how it forms. So whenever the uterus is forming, it starts out as 2 tubes and it'll fuse together and form the uterus. But in this case, it doesn't fully fuse together, and there will be a septum that remains and divides up the uterine cavity. So this will cause a very high incidence of miscarriage. But the good news is that it's easily treated by a hysteroscopy to surgically remove the septum. This will decrease the miscarriage rate from 85% to 10-15%, and it'll actually double the term birth rate from 10% to 20%, which is way better than it was before. You can obviously see in figure 1 the septum. There's a lag, isn't there? Okay. So the next would be Uterus Dophys. So this is very similar to the septate uterus in that it's a abnormality with diffusion of the 2 ducts or tubes, but in this case, it's a complete failure of the ducts to fuse together. So this will actually develop 2 separate uteruses, each with its own ovary and fallopian tube, and occasionally in rare instances, it'll actually form 2 vaginas. The problem that occurs though is that normally it's supposed to be 1 uterus, like a normal size uterus, but in this case it's 2, but they're only half the size. So pregnancy is very dangerous. So I interviewed Dr. Jackson for what his experiences with this, and he had a patient come in with uterus Dophys, and they had ruptured membranes very early at 22 weeks. They gave a normal delivery, but they had developed an infection from the ruptured membranes, which has to be treated immediately. The patient can become septic very quickly. So after delivery, they tried to remove the placenta. They had very difficult time due to the very small size of the uterus. As we said, it's only half the size it should be. They tried to use something called a suction cup technique, where they try to remove the placenta, but in this case, it did not work, and the patient then became septic and needed a hysterectomy. In the OR, Dr. Jackson he discovered that when he had opened her up, the placenta had actually grown into the uterus. It was stuck. Trying to pull it off can damage the uterus and cause the patient to bleed out on the table, which is very dangerous, especially with someone that's already septic. He very quickly saw the challenges of trying to delineate the abnormal anatomy of the cervix and uterus. So all the vessels are completely abnormal. There's two uteruses. That means there's going to have very different vessels going through there. So the patient was already bleeding out, and it was in septic shock. So there was no time. She was on 3 vasopressors, which is essentially it helps increase the blood pressure. He only removed 1/2 of the uterus Dophys, so he removed only 1 uterus, leaving the other functional one in. The mother actually made a full recovery, and because Dr. Jackson decided to only remove 1 uterus, she actually got pregnant in the other uterus and gave birth normally. Yeah. So the next system would be the cardiovascular system. More specifically, the first one is the ventricular septal defect. So this is actually a very common and often unnoticed congenital heart anomaly. It's a hole that connects the 2 ventricles of the heart, which allows oxygenated blood to pass from the left to right ventricle, which is then pumped back to the lungs. That's a problem because we only want deoxygenated blood being pumped back to the lungs. So the heart is doing unnecessary work. So this can result in some heart failure, pulmonary hypertension, endocarditis. A lot of problems can occur. No clear cause. So there's some like speculation, genetics, environmental factors can play a role, but there's no idea. Treatment, usually smaller holes will actually close on their own. The larger holes will require surgery, and sometimes medium-sized holes. But the next would be bicuspid aortic valve. So the cusps, there's supposed to be 3 cusps on the aortic valve, but in this case, there's only 2. It's present from birth. It can occur with many other heart defects, but this is actually a much greater heart defect than people realize. So after interviewing Dr. Jackson again, he had told me how the aortic valve, bicuspid aortic valve, can cause aortic stenosis, which is essentially the narrowing of the exit causing a fixed cardiac output, which is just the same thing as saying, like, you know, you're only going to get so much output from a fire hose. You can't increase that. So it's like if you are scared and your adrenaline's pumping and your body's asking for more blood, your heart's going to try to pump out more blood. But if it's a fixed cardiac output, it's only going to pump out the same amount it was before. Then you could have a drop in blood pressure. Not good. So this is especially a problem during pregnancy because cardiac output is normally supposed to increase by 50% and then another 50% during labor, whereas during systemic vascular system decreases and then the blood pressure is not going to be maintained. The heart pumps even harder, attempting to compensate, but fails to, and the output, because the output's fixed, and it's going to only use more oxygen because the heart's a muscle. The coronary arteries aren't going to get enough blood to oxygenate the heart, which is going to result in cardiac ischemia and ultimately death. Another thing to consider is that during labor, pain is actually going to increase the sympathetic output and increase the heart rate, which leads to a supply-demand mismatch because of the fixed output. So a local anesthetic, in an attempt to block the sympathetic output, will actually only decrease the blood pressure. But that's a problem because the heart's going to try to compensate for that by beating even harder than it was before, which is only going to drop the blood pressure even more. So the cardiac output would only decrease even more and ultimately cause death. So Dr. Jackson had a patient with a bicuspid aortic valve. So he induced her in the cardiac ICU, and they very closely monitored her for blood pressure while doing a narcotic epidural to provide some pain control, but not as much as the sympathetic block. It's kind of like they found the happy medium in between, where they slowly introduced a block, but not too much. They got the baby oriented in a position where they could perform a forceps delivery, which is essentially the same thing as taking medical pairs of forceps and pulling the baby out by its head. Very carefully, obviously. The next system may be the nervous system. So first one would be the spinal bifida. So this is essentially the neural tube defect in the development of the fetus. The bones do not form properly, leaving part of the spinal column unprotected. There are 3 types. Myelomeningeal, which is the spinal canal is open along several vertebrae, which allows membranes and nerves to push through, which form a sac. You can actually see that in figure 6. It's the third option. It's very dangerous. Then there's the meningeal, which the spinal canal is only open for a small number of vertebrae, which only allows the meninges to protrude through. And then there's spina bifida occulta, which is a very mild form, usually goes unnoticed. Usually doesn't require treatment either. So causes can be a combination of genetics, environmental factors, folic acid deficiency. They really don't know. It's only speculation, although it's very common. You'd think they would. Treatment, meningeal will require surgery. So the surgeon is just going to open them up, push the nerves and membranes back into the spinal column and close them up again. Sometimes they'll use a shunt to fill in the space where the vertebrae is missing. The next would be hydranencephaly. So this is a very, very rare cerebral anomaly, which the brain's hemispheres, the cerebral hemispheres, will not form. They'll be completely absent, and they're going to be replaced by sacs filled with cerebrospinal fluid. The birth will be completely normal. The baby will come out looking normal, but the problem is very quickly they will notice that the baby is not right because it'll continue to accumulate fluid within its cranium, and the cranium will increase in size. The pretty quickly, and it's not good. Causes, there's very little known because it's so rare. There's a lot of theories talking about that it's an inherited disorder, it's caused by a blockage of the carotid artery. No one really knows. Treatment, well, there's really no cure or treatment for not having a brain, so yeah. The next would be the muscular system, muscular dystrophy. This is a group of disorders that's characterized by progressive muscle weakness. So it's the key protein in this is involved with this is called dystrophin. It's going to play a role in muscle integrity during contraction. The problem is that a lack of dystrophin is muscular dystrophy, which can cause many problems with things like walking, swallowing, or muscle coordination. So it's caused by defects in the dystrophin genes, which reduces or eliminates the production of all dystrophin. So if you look at figure 11, you'll see that green chain looking thing that connects the F-actin to the sarcolemma, the membrane. So whenever there's contraction, the dystrophin will maintain the muscle integrity rate. But without dystrophin there, the distance between those two will increase, which will then cause a lot more weakness during contraction. There's no treatment or no cure, but there are some very promising drug treatments. The next, my favorite, myostatin-related muscle hypertrophy. This one's really cool because it's an extremely rare condition which causes like very reduced body fat, and you have essentially twice as much muscle mass. And of the muscle that you do have, it is twice as strong. There is literally no medical problems that they have discovered from this. So it's actually a good thing from what they've seen. Causes myostatin restrains skeletal muscle growth, and they actually have done quite a few studies on this, and mutations in the MSTN gene will reduce the production of the functional myostatin, which causes an overgrowth of muscle tissue. So if you look down there, figure 12, the Belgian blue cow, so there's some scientists in Belgium that got a little too carried away and played with the genes, the MSTN genes within some cows, and they actually created this really strange breed of cows, and they were absolutely enormous. And so you can actually see them because they have huge herds of Belgian blue cows. And it's kind of cool. And so you can actually see it in children very easily. So if you look at figure 13, you'll see that image of that boy. He obviously has a much larger muscle mass than the average toddler. Obviously, there's no need for a treatment. So my own personal experiences. So there was a lot of variation within the donor, but just here's a couple that I found were kind of interesting. So the first was the donor had a very large anterior jugular vein. So this normally varies and is inversely proportional to the external jugular vein. And it's normally supposed to be a paired vessel, so on either side of the neck. But the donor had actually a large single vein on the midline of the neck. So it's normally supposed to drain blood from the anterior aspect of the neck into the subclavian vein. So I can only speculate that there was a greater blood supply needed from the anterior aspect of the neck, and it could have been some severe thyroid issues, but I'm not sure. So there was actually these doctors that published a study on this where they were going to perform a tracheostomy to save this woman's life, but right before surgery, they stopped and scanned her neck, and they found that she actually had this rare anatomical variant, which caused her anterior jugular vein to cover her trachea. Had they not checked, the woman probably would have died on the table. And lastly, there is the dorsal scapular artery. It actually had originated very abnormally. It is normally supposed to originate on the thyrocervical trunk and pass superior to the brachial plexus, but in this case, the donor had it originating more distally off the subclavian vein or artery and passing between the upper and middle trunks of the brachial plexus. So that's just a group of nerves. And so they've speculated that it can act as a nutrient artery to the brachial plexus, trunks, and cords. It's actually it's not uncommon. So it's probably, it might be just due to evolution, not sure. But that's everything. Thank you guys. Are there any questions?
See, I have a question for you, Ben, actually. So I was wondering how you landed on each one of these abnormalities. Was this just kind of from a list that you found interesting, or were these ones that you found out from that doctor through your visit with him?
It's a good question. So I did a lot of research into each body system. So I kind of tried to keep it as like there was categories. So I kept it at like the most common, and then one that I found extremely interesting or super rare. And then I tried to find, talk to different doctors and find their experiences. And that would have been my third one or second one in this case, in some of the cases. But it was pretty easy to find the most common one. But the rare ones were very difficult. So I ran across a ton of really cool ones, but obviously there's a pretty limited space on a poster. But yeah, did a lot of research. Looks like there was a question in the chat. Yeah, it said, what was your favorite or most interesting anatomical abnormality that you discovered during your research? Yeah, so since a lot of them didn't make the cut, there was one of them I found very interesting. It's called fetus in fetu. It's basically where during the normal development of twins, the zygote it's supposed to split off and form two separate fetuses. Except in this case, it doesn't fully split off, and still part of it is connected, the two zygotes. So over time, one fetus will overtake and envelop the other fetus, and it doesn't normally when that happens, it'll take in their chromosomes, it might express it. That's why you see two different eye colors on some people or stuff like that. But in this case, it doesn't absorb it at all, and that fetus will continue to develop within the other fetus, but at a much more slowed rate. And it's non-viable, but it's still super cool, because you can be like there could be like a 6-month-old baby that has like this small like orange sized clump within its abdomen of this like this fetus like mass. Or like they actually found a 30-year-old with a fetus in fetu, which I'm amazed that they went this long without discovering that. And they found he actually had it develops these like actual characteristics of a baby. Like there's one with like feet, hands, like one started developing a heart, like hair, teeth. Yeah, it's really interesting. How often is atypical anatomy discovered prior to surgery through imaging, and how often is it discovered due to surprise during the medical procedures? Actually, it's really rare that they discover it during the procedure, because there is a lot of prep that goes into it, because you can't anticipate that kind of thing. So it's kind of standard procedure that they do a lot of imaging before they go in, because they don't want to be ill prepared. And oftentimes, they're going to see symptoms that indicate certain abnormalities, so they can probably walk through the procedure having a pretty good idea of what's going on, along with a lot of imaging. Do you know if twins are more likely when a double uterus is present? No, actually no, because usually, maybe it might start out like that, where each uterus might have its own baby, but over time, since they're only half the size of the other, they're it's not enough space. They might develop over time, but they're both going to inevitably pass, because there's not enough space. The ruptured membranes will happen way too early. It's really dangerous. Is there any other questions?
All right, if there are any more questions, feel free to just kind of type them in the chat. I think we'll have a little more time at the end of the presentations. Otherwise, Ben, do you want to go ahead and switch the host power?
Yeah.
Okay, sorry about that. So next up, we're going to have Valerianna Gentzsch and Sydney Wisnefski, who are going to talk a little bit about antibiotic-producing bacteria that they discovered while working in a microbiology class. This was 3 semesters ago, I think. So they didn't have a chance to present this during last year's Undergrad Scholars Forum due to COVID, so they get to do it this year.
Okay, can everyone hear us? Okay, hi guys. So our presentation is over bacteria associated with antibiotic production and resistance. This project was performed, like he said, in the fall of 2019. And then, so to get started, antibiotics are molecules which inhibit the growth or kill microorganisms. The discovery of antibiotics is arguably one of the most important discoveries in medicine, as they have allowed us to overcome simple infections which would have been considered deadly in the past. So although antibiotics are very effective, recent years have seen a spike in antibiotic-resistant bacteria. This happens when mutations accumulate and the bacteria is no longer affected by the drug used. When treated with the antibiotic, the drug-resistant bacteria becomes more abundant than the normal bacteria. So in figure 1, this process is where we explain this. The orange bacteria is the normal bacteria and the blue is the resistant bacteria, and then the tan bacteria is the dead normal bacteria. And so as you can see, the blue resistant bacteria has taken over. The emergence of antibiotic-resistant bacteria creates a major problem in clinical settings, as simple infections can become quite difficult to treat. One major area of research is the identification of new antibiotics. One place to look for new antibiotics is in soil microorganisms. Here, bacteria often create and secrete antibiotics to kill off their competition. So the goal of our experiment is to isolate new antibiotic producing bacteria from soil. And we collected our soil from Westminster College's quad. Okay, so to begin our experiment, we had to isolate antibiotic producing bacteria. And so we did this by obtaining a soil sample, and then we diluted it with sterile water and put a thin layer of this soil water solution onto a plate. After the bacteria grew, we then chose 10 bacterial colonies we thought might produce antibiotics. So we chose colonies that thrived in growth compared to surrounding colonies. So from here, we gram stained our 10 bacterial colonies. So we chose to gram stain the colonies to see which type of bacteria, either gram positive or gram negative, like which ones the antibiotic resistant would affect or kill. So for our initial test for antibiotic production, we chose to perform a cross-streak assay. So in figure 3A is a figure representing the method where we streak the bacteria we are testing for antibiotic production vertically down the middle. And then we test this bacteria against other bacteria colonies from our soil by streaking those bacteria horizontally across the plate. This results in 2 outcomes. So the top line represents the bacterial growth is not inhibited. This means that our bacteria does not produce any antibiotics. The bottom horizontal lines represent bacterial growth being inhibited because the bacteria cannot grow close to the bacteria we are testing for antibiotics. This shows that our bacteria produces antibiotics. Figure 3B shows our actual tests, and on AP4 plate, which is right here, the bottom horizontal line represents bacterial growth being inhibited and indicates the vertical line of bacteria produces antibiotics. And then the horizontal line right above it indicates that the bacterial growth is not inhibited by the vertical bacterial streak. So once we determined which bacteria produced antibiotics, we aimed to purify the antibiotics. And so we did this by centrifuging the bacteria, which is a process of spinning down the cells to strengthen the antibiotic. So you can see in figure 4, it shows the purified antibiotic in a microtube. And so we kept the supernatant, which is the top layer, for further testing, and discarded the pellet, which is the bottom piece. And then to test the supernatant for antibiotic properties, we covered the entire plate with a gram-positive bacteria. So we used Bacillus megaterium and placed small filters soaked with supernatant on the lawn of the bacteria. So you can see that's the little like white circles are the little filters. So this is shown in figure 5, as Val pointed out. As you can see around isolates 1, 4, and 8, the area around the supernatant has this zone of clearance. So that's like this small area where nothing is growing.
and it suggests that they killed the bacteria. And this conclusions table is what is like shown in figure 5. So although we tested our supernames on multiple other bacteria, 1, 4, and 8 were our strongest results. And since we only had 1 semester to work on this project, we were not able to perform as many tests as we would have liked. But if we continued our research, our next step would be to further purify the potential antibiotics and test their ability to kill other bacterial species. Is there any questions?
All right, so I'm gonna talk loudly so that way we don't get echo. So I did have a question. What was maybe one of the biggest things you learned just about the research process in general? I know it was a long time ago, but if you could just kind of talk about that a little bit.
So it definitely doesn't go how you think it's gonna go. We had lots of like issues like with like our bacteria like wouldn't grow, so we'd have to like start over, or just like the results we thought we would get, we didn't get, so we kind of had to change our pathway. There were just a lot of different factors that we like didn't expect, so you had to work around a lot of things.
Yeah, and I think that sometimes if you don't get the result that you think you're gonna get, that's still a result, and like you can still go from there and get info from there. There's a question in the chat. Do different soil types have different antibiotic activity levels? I think so. So each group like got them from somewhere around Westminster, so it could have been anywhere. So ours was just in the quad, and actually it was kind of gross how many things were growing in the quad, but each group did it from somewhere else, and each one had different and like different things produced from that.
Yeah, we even had fungus growing.
Yeah,
not just bacteria. So
does it matter how deep the soil was? Surface or lower? I don't think that it matters what.
yeah, I don't really remember how. I think we just got surface levels.
Yeah, I think so
I
too.
don't think we paid attention to how deep it was when we got our soil. So.
Let's see, any other questions? All right, so we actually still have about 20 minutes. That can kind of end the presentations, but if anybody just wants to kind of chat about how research is done in general, or wants to talk about these posters in a little more detail, feel free to kind of stick around and ask a little bit more questions. Okay, if there's no other questions, I think we could probably go ahead and call it a little early. Feel free to jump in on another session. There's a few others going on at the same time. Otherwise, yeah, feel free to email or anything if you have more questions.